So, so we are at the meiosis, right? The production of offspring by sexual reproduction includes the fusion of two gametes, right? Each with a complete haploid set of chromosomes. Now, gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half results in the production of haploid daughter cells. Now, this kind of division is called meiosis, and meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, whereas fertilization restores the diploid phase. Okay, so meiosis makes the haploid phase, and fertilization makes the diploid phase. Okay, there are gametogenesis in plants and animals, that is formation of gametes. This is the meiosis. Okay, this leads to the formation of haploid gametes. The key features of meiosis are as follows. Now, meiosis involves two sequential cycles of nuclear and cell division called and then cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Okay, only a single cycle of DNA replication. Now, meiosis 1 is initiated after the parental chromosomes have replicated to produce identical sister chromatids at the S phase. Now, meiosis 1 is initiated only after the parental chromosomes have replicated. Chromosomes have replicated to produce identical sister chromatids at the S phase, right? Now, meiosis involves pairing of homologous chromosomes and recombination between non-sister chromatids of homologous chromosomes. Now, 4 haploid cells are formed at the end of the meiosis 2. Now, meiotic events can be grouped under the following phases: meiosis 1, meiosis 2. Now, prophase 1, metaphase 1, anaphase 1, and then telophase 1. Same as meiosis 2, prophase 2, metaphase 2, anaphase 2, and then telophase 2. Now meiosis 1. Now prophase 1. Prophase of the first meiotic division is typically longer and more complex. Prophase of the first meiotic division is typically longer and the more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on the chromosomal behavior. Now, based on the chromosomal behavior, it is of 5 phases. First one is the first. So the first one is the so the first one is the leptotin, then zygotin, then packaging, then diplotin, and then dikinesis. Now, during leptotin stage, the chromosomes become gradually visible under the light microscope, right? The compaction of chromosomes continues throughout leptotin. This is followed by the second stage of prophase 1 called zygotin. Now, what happened during the zygotin? The chromosomes starts pairing together, and this process of association is called synapsis. Okay, this process is called the synapsis. Now, such paired chromosomes are called homologous chromosomes. Now, electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of complex structure called synaptimal complex. So, first synaptimal complex, then synapsis occurs. That is, they start pairing together. Okay, start pairing together, right? So this is it. So, synaptomal complex. The complex formed by a pair of synapse homologous chromosomes is called bivalent or a tetrad, because there are 4 chromatids. So they are tetrad. Okay, bivalent. Okay, so just remember the term that is started pairing, that synaptomal complex, then bivalent or tetrad in the zygotin stage. However, these are more clearly visible at the next stage. I mean, these bivalent chromosomes, they are more clearly visible in the packaging. That is, the first 2 stages of the prophase 1 are relatively short-lived compared to the next stage, that is packaging. During this stage, the 4 chromatids of each bivalent chromosomes becomes distinct and clearly appear as tetrad. Okay, now tetrad. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Okay, the recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between 2 homologous chromosomes. Now, crossing over is also an enzyme-mediated process, and the enzyme involved is called the recombinase. Crossing over leads to the recombination of genetic material on the 2 chromosomes, right? Recombination between homologous chromosomes is completed by the end of the packaging, leaving the chromosomes link at the sites of the crossing over. Now the beginning of diplotin is recognized by the dissolution of synoptimal complex. You can remember as D for diplotin, D for dissolution of synoptimal complex, and the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of the crossovers. Okay, and they show the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of the crossovers. Okay, now these X-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. Now in oocytes of some vertebrates, oocytes, that is the stage in the meiosis during the formation of gametogenesis, oocytes of some vertebrates, I say diplotin can last for months or years. Now just remember diplotin dissolution and then X-shaped structure called chiasmata. Now and this also line remembers that leaving the chromosomes linked at the sites of crossing over occurs at packaging. Okay, this also remember, like in diplotin, the tendency of the recombined homologous chromosomes of the bivalents to separate from each other except at the sites of the crossovers. Okay, so these X-shaped structure are called chiasmata. Now the final stage of the meiotic prophase 1 is dikinesis. This is marked by the terminalization of chiasmata, right? Now during this phase the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation, right? So by the end of the dikinesis, the nucleolus disappears and the nuclear envelope also breaks down. The dikinesis represents the transition to metaphase. Now metaphase 1, the bivalent chromosomes align on the equatorial plate. The microtubules from the opposite poles of the spindle attach to the kinetochore of homologous chromosomes, right? Now the anaphase 1, the homologous chromosomes separate while sister chromatids remain associated at their centromeres. This is not in the mitosis. The centromere gets split, but anaphase 1 it does not split the centromere, right? This occurs in further like anaphase 2. So telophase 1, like the nuclear membrane and nucleolus reappear. Cytokinesis follows. The cytokinase follows, and this is called as dyad of cells, right? Division of cytoplasm, then dyad of cells. Two cells are formed. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called interkinesis. Two meiotic division, right? Interkinesis, and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis is followed by the prophase 2. Okay, a much simpler prophase than prophase 1, right? So this is prophase 1, then metaphase 1, then anaphase, and then dyad of the cell. Like two cells are formed, right? Two cells are formed, as you can see. There is a crossing over between prophase 1. There is crossing over between prophase 1. Then there is then there is metaphase 1. The chromosomes are crossed. Okay. So as you can see, like if it's so you can see. Okay, so yeah, you can see in the diagram there are like double copies of chromosomes in each set, you see, and they gets reduced. Like you can see there are like total 8 chromatids, right? Now in the progeny itself, one daughter gets 4 chromatids, so you can see they get reduced. So this is why it is called the reduction division. Now the meiosis 2. Now prophase 2, meiosis 2 is initiated immediately after the cytokinesis, usually before the chromosomes have fully elongated. Now in contrast to meiosis 1, meiosis 2 resembles a normal mitosis. Okay, in contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. Okay, I mean the nuclear membrane disappears by the end of the prophase 2. you know what I say, the nuclear membrane disappears by the end of the prophase 2, same as during the prophase 1. The chromosomes become compact. The chromosomes again become compact. The metaphase 2. At this stage, the chromosomes align at the equator and the microtubules from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Okay. Now anaphase 2. It begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell by shortening of microtubules attached to the kinetochores. Right. This is the anaphase 2. Now this is prophase 2, metaphase 2, then anaphase 2, and then it is telophase 2. You can see dyad of the cells get now tetrad of cells form, like that is 4 haploid cells. So telophase 2, meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is 4 haploid daughter cells are formed, as you can see. If it's 2n, then they are also 2n. You see here it is okay, but the chromatid, you can see it is 4 chromatids, so it is 2 chromatids. This can be also asked, like in prophase 2 there are 4 chromatids, as you can see, but in prophase 1, in telophase 2 there are 2 chromatids. You can count the chromatids. So but the copies are same. Here it is 2n, here it is 2n, but in the first diagram, as you can see, here it is 4n. Just say 4n. Now it is how much? It is 2n. I mean it is reduced. It is reduced, right? It is reduced. So it is reduced, right? Okay. And the significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organisms. Even though the process per se paradoxically results in the reduction of chromosome number by half, it also increases genetic variability in the population of organisms from one generation to the next, and variations are very important for the process of evolution. So this is the significance of meiosis. I read it again. Meiosis is the mechanism by which conservation of specific chromosome number of each species is achieved across generations in sexually reproducing organism. Why is the conservation of chromosomes? Because you see, if they get reduced, then next after the fertilization they get diploid, and again then get reduced. So it's type of like conservation. So meiosis doing the conservations, right? It conserving the chromosome number. Even though the process per se paradoxically results in the reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variations are also important for the evolution. So variations are the key part of the evolution. If there is variation, there is the evolution. Now this is your chapter, and there is a summary, and this is it. You are done. Again I can explain you some like diagrams, is if you because these diagrams are very important. Prophase 2, then metaphase 2, then anaphase 2, then telophase 2, and here it is. Prophase 1 has like just remember this diagram, right? This diagram is very important. Just see, just remember that there are 8 chromatids. Okay, there are 8 chromatids. Now they gets like 4 chromatids in each daughter cell, so it gets reduced. And also it says, like here is the line that meiosis 1 is initiated after the parental chromosomes have replicated. So in meiosis there is the replication of parental chromosomes to produce identical sister chromatids at the S phase, right? Now this is the interphase, this is telophase, anaphase, this is mitosis, metaphase, then transition to metaphase, late prophase and early prophase. So yeah, this is it. Like here is also the splitting of the centromere occurs in mitosis, right? Now Now this is it, this is your chapter is done. Okay, so this is it.